Sexual selection has led to the evolution of sometimes bizarre traits in nature. Describing the evolutionary mechanisms behind intersexual competition, or female choosiness, is extremely difficult. My paper is titled, Meta-Analysis Suggests Choosy Females Get Sexy Sons More Than Good Genes. The authors are listed below on this slide. My journal was Evolution, and my paper was published the 10th of April in 2012. In class, we discussed two models of intersexual selection extensively. The Fisherian model, also known as the sexy sons hypothesis, is where females choose their mates because they are more attractive. This provides an indirect benefit for the female, because the sons of choosy females will possess these sexy traits and have higher reproductive success. The good genes hypothesis occurs when females choose their mates based on attractiveness that correlates with the honest genetic quality of their mate. The offspring in this hypothesis will have traits that make them more fit. There is much debate in the research community on the quality of evidence for both these models and whether or not one occurs more often than the other. Understanding the merits behind each model is extremely important. This paper aggregated information of 90 studies in order to evaluate the empirical evidence for the sexy sons and the good genes hypothesis. The study answers the question, is there significant empirical evidence supporting both the Fisherian model and the good genes model? In other words, the hypothesis is that there will be empirical evidence that sexually selected traits will be associated with offspring fitness in terms of either sexual attractiveness of sons or other fitness components. This paper analyzed 3 categories of fitness traits and measured the trends these traits had on indirect benefits or offspring success. First, sexually selected traits or attractiveness were tested. For example, this includes ornamental traits. These traits support the Fisherian model. Second, life history traits were tested. Life history traits include survival, reproductive, or growth-related benefits. Third, performance traits tested for behavioral, size, and physiological benefits. Life history traits and performance traits provide good evidence for the good genes hypothesis because the benefits are honest indicators of male genetic quality. Now let's talk about how this analysis was done. Before finding studies that observe sexual selection, certain restrictions were placed. Studies with pseudoreplication, or faulty statistical methodology, were removed. And studies that did not have paternity for offspring that had been genetically confirmed or experimentally controlled were also removed. The final data set consisted of 90 studies with data on 55 species. The Fisher's Z-statistic was used to measure effect size. Effect size illustrates the magnitude that traits have on offspring success. This table explains what this statistic means more. The larger the value for ZR, the larger the effect size. Or in other words, the more the offspring benefited from the trait. Now let's talk about the results of this study. The y-axis on this graph is the effect size. The white square measures the mean effect size, while the error bars represent the 95% confidence intervals for the results and account for any variance. On the x-axis, you can see the 3 fitness traits that were tested for: life history traits, performance, and sexually selected traits. On this next slide, you can see more clearly what the ZR values are. Sexually selected and performance traits were associated with significantly greater effect sizes than life history traits, which had a ZR around 0. The significance of these ZR values might make more sense when compared to this table again. Even with the confidence interval, the 0.414 ZR value for sexually selected traits is significant, while performance traits also show a less significant but positive effect on offspring success. In conclusion, the study provides no support for good genes effects in terms of improved life history parameters in offspring. Interestingly, there was support for the good genes hypothesis in terms of a positive correlation between attractiveness and performance traits. Importantly, our results provide evidence for Fisherian benefits of mate choice. This result is especially surprising as it seems to suggest that evidence for the good genes hypothesis is ambiguous, even though many researchers support this hypothesis. Genetic studies such as this can be very helpful because they can allow you to holistically evaluate evidence for models of sexual selection. Despite this, it is important to understand that this meta-analysis does not disprove methods for intersexual selection. Instead, it measures the significance of available data. A small number of studies analyzing such relationships make it difficult to draw more definite conclusions. There are also some potential pitfalls with meta-analysis studies. For example, there may be a bias to find a particular result or a manipulation of data. Overall, these studies could benefit from more data and from a greater understanding of other factors that impact models of sexual selection. Let's try to understand this in terms of some other studies. First, let's look at a similar meta-analysis, which had a smaller sample size of only 22 papers and 22 species. They found only a slight positive correlation for life history traits, specifically survival and offspring success. These were very different results. This study had limited information, which illustrates that the more data you are able to gather, the more the trends will become apparent and the more significant your results become. The zebrafinch study is a great qualitative example of the Fisherian hypothesis. Zebra finch mates are sexually selected for a color ornament that has been exaggerated, even though it could warn predators of their existence. This study also illustrates a confounding factor for the Fisherian model. Differential maternal investment can actually affect the color of the beak. It may not be related to the male's genetic data. Poison dart frogs are a good example of a study supporting the good genes hypothesis. Calling rates in males are a genuine indicator of their genetic quality, as females that mate with better callers have higher quality offspring that are more likely to survive. I included this example we studied in class because it is important not to conclude that good genes don't have an effect on offspring success, just that with the existing data this study had, there was an ambiguous statistical correlation. Thanks for watching!